Okay, so this is going to be the last video of the semester. I'm going to be cramming in all parts of adaptive immunity, and I'll be calling it a day after this. And so in the previous video, we talked about the differences between innate immunity and adaptive immunity. We talked about such things as the differences between passive and active immunity. That's kind of important. We've been living through the horrors of a pandemic. We've been dealing with the all the drama surrounding the immunization program that has successfully, I'd add, stemmed the spread of COVID-19. And we're going to be mostly talking today about two things, right? There's the top half of the slide and the bottom half of the slide. The top half is just telling me that I needed to kind of briefly explain, to walk you through the process of adaptive immunity and what is going on. How does adaptive immunity help you out when you are being under attack by some type of pathogen, right? And then the slightly, actually, substantially more complex bit of the story is going to be on the process, the mechanism by which we generate an adaptive immune response. Okay? And it's going to seem kind of detailed and fidgety, but I promise you that it just gets worse later on. I mean, it's such an involved process, but it's also incredibly important. So I'm sort of visualizing this as maybe your first dip into the pool of immunology discussions that will be part of your life for as long as you remain a biology major, okay? Or an allied health worker. Okay, so let's go ahead and get started. Now, I told you that there are going to be two sides to adaptive immunity. You've got the humoral immunity, or the antibody-mediated immunity, which distinguishes it from the cell-mediated immunity, and basically reminds us that in the case of cell-mediated immunity, we have cells. Right? We have cells, whereas with humoral immunity, we have cells that produce the proteins, but it's actually proteins that are the things that confer immunity to the person who's immune, right? And so these things, we call them antibodies. You can also call them immunoglobulins, right? They're proteins. And as we've learned very recently, if you're a protein, you must have come through a process of translation, which in turn was made possible because of transcription of genes. And so, yeah, we've got these genes in a population of cells called B lymphocytes. B cells. And these B cells, during embryonic development, they do something that's really kind of crazy, spectacular. They undergo an enormous proliferation and diversification such that each B cell that comes out of this makes a different kind of antibody. Only one kind. It's got the genes that are able to make the mRNA molecules that are able to be translated, and the protein that comes about is going to look like this. Antibodies are always kind of shaped like Ys, like the letter Y. You've got two long chains; these are called the heavy chains, and you've got two short chains. It's called the light chains. And on this molecule, on the arms, right, where you've got the end of a light chain and the end of a heavy chain, you've got two of them. These correspond to the antigen binding sites. Okay. And what these antibodies do is that they bind to an antigen. And remember, an antigen is the thing that I called in the previous video, the mark of the enemy. So the thing that we're able to use to recognize a pathogenic agent as not being part of yourself. In other words, it's important to recognize self from non-self, but it's really important for you to be able to tell a particular kind of thing that is going to be dangerous. Okay? It's going to cause an infection. And those are the ones you want to generate an adaptive immune response against. Okay? Now, the enemy, it could be a virus, it could be a toxin, could be a bacterium, could be a cancer cell, could be even one of your cells that's been virally infected. Okay? And all of these things are going to be bearing antigens that are going to be recognized and addressed through adaptive immunity. Okay? These last two, cancer cells and virally infected cells, are probably going to be in the domain of cell-mediated immunity. But viruses, viral particles, toxins, and bacteria, things that are small, are probably going to be addressed with the antibodies. Okay. These immunoglobulins. And so what the what this little Y-shaped molecule can do is it could bind to a toxin. Right? If it, so in other words, if the antigen is a toxin, it's part of a toxin, it can neutralize the toxin. Basically, make the toxin no longer effective at binding to whatever the toxin does and causing the mischief that it does, right? Some antibodies are called bacteriolysins. They actually will cause the bacterium to break apart. And so bacteriolysins is like a direct killer of the bacteria. All the antibody has to do is to bind to the bacterium and it lyses. Okay? You can have the virus, the antibodies serve as, we call them agglutinogens, because they cause little things like viruses to agglutinate, right? So you're infected by a virus, okay? You got these viruses, thousands of viruses all over the place. That represents a huge risk because those viruses are able to go out and find host cells to infect, right? Now, if you could agglutinate them, if you could take all these viral particles and make one big gigantic gob of viruses, okay? Then those viruses cannot go out and attack other cells, and along comes a macrophage, and a macrophage can come along and engulf it, right? So these are all ways to make yourself better simply by producing the appropriate protein that will bind to and do its thing with the pathogenic agent, right? So humoral immunity, yay, right? Yeah, it's like when you get the flu vaccine. What you're doing is you're training your immune system. It's an active immunity, right? So you're training your immune system to generate antibodies that will be specific against the viral antigens that are present in that vaccine. Okay? So because those antigens are formulated to be the same as the antigens on the actual flu viruses that are going around, they're circulating that year, you automatically gain immunity from the flu vaccine. Okay? Yeah. Sometimes the people that generate the flu vaccine don't hit it exactly right. So in other words, they might use antigenic determinants from strains of the flu that are different from the one that actually becomes the epidemic strain, right? In which case, people get the flu vaccine, but they get the flu anyways. But on the whole, most of the time, the flu vaccines are pretty good because we've gotten pretty good at predicting which flu strains are going to be causing the epidemic in our area. And moreover, if you're immune to even the wrong kind of flu, that confers some degree of immunity you against the flu version that comes through. You might still catch it, but it likely won't affect you as badly as someone who has no flu immunity at all. Okay, so you've probably heard that story before, but I thought I'd throw that in here. Okay, now the other kind of immunity is cell-mediated. And so what I want to do here is to kind of remind ourselves we've got a different population of cells. We've got, here, you've got these, I think your book calls them defensive T cells, right? But I'm gonna call them cytotoxic T cells, TC cells. Right? And what the cytotoxic T cells do is that they destroy and they'll locate cells that bear the signals, the antigenic marker, the mark of the enemy. And again, these might be your own cancer cells, they might be viral infected cells, but they'll simply just kill them. Right now, let me show you a picture of that. And so go to my images from the textbook. And here we go. This is a cytotoxic T cell. And what's happening is that the T cell is recognizing two things. It's recognizing the foreign antigen. I'm not terribly thrilled with this picture, but I'll pretend like it's okay. You've got the foreign antigen, and you've got the stealth marker. And the cytotoxic T cell requires a recognition of both things. And what happens is that when cytotoxic T cell is activated, it will basically, when it's activated, when it recognizes a bad guy cell, it's the bad guy cell. Okay. Remember, it's either virally infected cell or a cancer cell. It will basically spray the cell with a can of mace, right? Effectively poking a hole in it. There are perforin, and you've got granzymes. Okay. These things that are inside the cytotoxic T cell are released from this T cell. They'll actually form a hole in the bad guy cell, and the bad guy cell is then destroyed, and it turns into this dead cell, right? And so the way that cytotoxic T cells work is by directly killing the virally infected or cancerous cell, and that's different from the way that antibodies work. Antibodies, if you remember, are simply going to bind to the pathogenic agent, and by binding to them, that's sometimes that's simply enough. Right? We can cause the viruses to agglutinate, we can cause the toxins to neutralize, we can cause the bacteria to lyse, and sometimes we might simply just attach to things that can be then more easily recognized by the other cells of our immune system, both the innate and the adaptive. Right. So if you've got generic macrophages and phagocytes out there looking for things that don't belong, right? It comes across something that is completely covered by antibodies. It's going to say, "Hey, that thing over there that's covered with antibodies, we need to get rid of that thing," right? And so the antibodies can certainly be of some help in that way as well. Okay, now back to my whiteboard share. We've talked about the ways in which immunity can work. Good with there, check. Now you know what happens when you have launched one of these massive counterattacks against a very specific pathogenic agent, right? And now the next thing, the second half of this lecture is going to be talking about what happens. How do we generate that adaptive response? And you know, it's not an easy lesson, and it's not an easy thing at all for our immune system to do. Right? It requires lots of parts, and because it requires lots of parts, it's actually kind of, it's kind of parallel with the the effort that your immune system has to go through in order to generate an adaptive response. is very similar to the effort that you're going to have to do to basically to learn the basics of how this works, okay? Because this turns out to be a pretty important lesson in your physiology and microbiology classes, which is why I'm asked to prime the pump, as they say here, to kind of get you thinking about the kinds of cells that are there in your body that will take part in this very involved process of launching and generating an adaptive immune response. All right, so let's go. And in the previous lecture, we already talked about these guys, the antigen presenting cell. Okay? In fact, I drew you a picture, those dendritic cells, those cells with long little arms. They phagocytize bad things. They're actually they start working very early in the process. At the same time that inflammation is going on, these dendritic cells are encountering the pathogens, and they're starting to prepare them for activation, for activating the T helper cells, right? And so these guys will then turn over, will combine the self non-self complex to a T helper cell. Okay? These are the CD4 cells. Okay? These are the different population of cells, and then the T helper cells, they're the ones that go out to activate the B cells and the cytotoxic T cells. In other words, remember I said at the beginning, we've got a whole bunch of B cells. They're like sitting around, ready to go, but not really ready to go. They need to be plugged in. They need to be turned on, right? You can't turn them on simply by attaching to the antigen, right? It's like, yeah, they need to encounter the antigen, but they also need to be authorized to move forward, or they need a special manager to come along with their little authorization key and plug in and then say, okay, B cell, it's time for you to step up for duty, right? And that's the thing that's required. That's the thing that takes a lot of time, which is why you don't launch an adaptive immune response immediately. It takes a few days for the immune response to launch in, and hopefully it happens soon enough for you to get better, right? Okay, let's talk about this thing. The antigen presenting cell, I've got a picture of this for you. Here we go. Okay, so in your textbook, I remember your textbook doesn't like to talk about dendritic cells. Macrophages are also antigen presenting cells, right? Our APC. Macrophage, a dendritic cell. And some B cells as well. Might as well be complete. B cells. Okay. There are a number of different cells that can present antigens. It's just that dendritic cells are like particularly good at it. Macrophages are kind of like multi-purpose. B cells are multi-purpose, but dendritic cells are kind of like they've got one thing that they do in life, which is to encounter bad things, take the antigens, and combine them with the self protein. The self protein is the MHC, okay? Major histocompatibility complex, and the MHC self complex. This self non-self complex is then passed along to the helper cell. Okay, this is the thing. This is the first handshake that's required in order to launch the immune response, right? The helper cell is not going to be able to do a whole lot, at least directly, to assist with the infection, right? It's not going to be able to produce any antibodies. It doesn't do that. It's not its thing. It's not going to be able to locate bad cells and destroy them. That's the CD8 cells. job, right? What the helper cell does is it goes out, this is the manager. It's got the authorization. It's going to be able to activate the appropriate B cell or cytotoxic T cell clone that can then go out and become the saviors, the superheroes that come along and help you get better, right? Okay, but this helper T cell, this is like the go-between. This is our required go-between, because those B cells and T cells, they cannot be activated directly. They need to be activated by an appropriate T cell who's carrying the antigen as well as the appropriate authorization code. All right? Okay, let's see where we are with this. Okay, so we know that we've got these antigen presenting cells, and they are going to be activating the helper T cells, and helper T cells then go out and will either activate the right B cell clone, that's for antibody mediated, for humoral, or the cytotoxic T cells, that's for cell mediated. And it's going to require the authorization through the helper T cells. All right. Now let's look at the textbook one more time. Last picture here is that once we actually find the appropriate cell, okay now there are so many metaphors that get used here. One of my favorite ones here is Cinderella. Everybody knows the story of Cinderella and how at some point she was the only maiden in the entire kingdom who had the right foot that would fit into the glass slipper. Right? Really? Okay, but that's the story. I know, but just bear with me here, okay? So you've got just one B cell out there in a kingdom that's full of other B cells. Only one of them is going to be able to make the appropriate antibodies to be effective against the invading pathogen, right? And so this helper T cell is kind of like the Duke that's got to go out and locate that one maiden in the entire kingdom, one B cell in the entire body that's able to do the right thing. And then once it's activated, this is the activation. Okay. The B cell starts to proliferate to make many B cells, right? So that one B cell starts to make copies of itself. It clones itself into thousands, tens of thousands, hundreds of thousands, right? So we end up with a whole bunch of these B cell clones that are going to be doing two things. One of which is to differentiate into these effector B cells. These are the ones that make antibodies in massive quantities, right? So they make the antibodies. They are, this is where all those antibodies are coming from. So you call them effector cells because they're effective right now. And then you've got the memory cells that also differentiate, and they don't really produce antibodies themselves directly. But what they do is they kind of stay along the outskirts, and they just bide their time. They say, well, I'm not going to produce any antibodies this infection, okay? But if I'm ever needed again, there are going to be plenty of us to become activated really quickly, as opposed to having to force the Duke of the T helper cell to find the appropriate B cell clone, one in the entire kingdom. There's going to be a whole bunch of us around, and we'll be able to become activated much more quickly than we did the first time. See that? Okay, so you get two things. We get the memory. This is why immunity lasts. This is why we can become, well, we can remain actively, potentially protected against pathogens for our entire lifetimes. Okay, sometimes. Okay, as we know, some immunities last longer than others. Okay, but in the ideal situation, the immunity will last forever, maybe decades. Okay, and that's by virtue of these memory cells. Okay, the effector cells. So, what's actually produce antibodies? They're there for only a short duration. Okay? They live fast and make a boatload of antibodies, but after they do that, they don't stick around, which kind of makes sense, right? Because you don't need to have a ton of antibodies produced forever and always. You're going to need to have the antibodies until your the pathogenic agent, your infection, is cured, and then you can stop, right? Okay. Now, a couple of things. These antibodies, they stay in circulation for quite a while. When you actually have one of these immune responses, you become highly immune. You start off with a high titer of the antibodies, but then, as we know, the effector B cells, the producers of the antibodies, they die, and then over time, months, years pass, that population of proteins in your bloodstream is going to start to dwindle, kind of falls off exponential decay. So you might start off with a super high titer of antibodies when you become immune, but the antibodies are going to be dwindling. Eventually, you reach the point where you don't have enough antibodies to protect yourself again, right? But that's not that big of a deal because you still have the memory cells. The memory cells last for much, much longer than this. Okay. So let's review here. Okay. You've got these antigen presenting cells, the APCs. Okay. APCs activate the T helper cells, and the T helper cells activate either the B cells or the cytotoxic T cells, B cells in the case of antibodies, cytotoxic T cells in the case of cell-mediated. And once activated, these guys become either effector cells producing lots of antibodies or immediate cytotoxic T cells, or they become memory cells. I'm not doing anything right now, but holding on and sticking around just in case they're going to be needed in a future infection. Okay? So now we can check off this part. Okay? The last couple of things are super important. I mean, it's important to ask questions like why, right? And so the first one is actually pretty easy. They're not all that hard, okay? But you know, first thing you might think is, well, this whole process takes days to effect. I mean, in that time between when the antigen presenting cell is first encountering the pathogen and the time that we actually get an adequate adaptive immune response can be several days, okay? During which time the infection could get really, really bad, and you're going to be suffering during that time. It depends on how aggressive the infection is. Okay, so in other words, by the time you actually have an immune response, you're already pretty sick. Now, you might say, well, that could be short-circuited if we just got rid of this middleman, right? If we got rid of the T helper cell and allowed either the antigen presenting cells to activate the B cells directly, or maybe the antigens themselves I mean, we've got these B cells. These B cells can be activated by the antigen, at least in theory. But no, the B cells require not only the antigen, they need the authorization of the self-protein that's presented by the T helper cell. Right? Why not make these B cells activatable without any required authorization code? Well, you can see the problem, right? We've got bazillions of different B cells. Each one has the capability to launch these enormously expensive immune responses, right? And your body doesn't have unlimited resources. You can't afford to generate antibody-mediated immune responses against a pathogen that's not really going to challenge you, right? I mean, you kind of do that in the case of vaccination, but you do that deliberately because you know that you want those antibodies on hand in case you ever encounter the flu virus or the SARS-CoV-2 or whatever it is you're becoming immunized against, right? So we're able to I think the T helper cells, to me at least, I think of them as being kind of like a necessary delay. It's like that second authorization. It requires two handshakes. Okay? First handshake is the one with the antigen presenting cell. The second handshake is where the T helper cell is going to locate the B cell. And the combination of those two handshakes is kind of like two-factor authorization. You don't launch something without the appropriate need, without the appropriate authorization. Does that make sense? Okay. The second thing to note here is that HIV, human immunodeficiency virus, is so devastating because it pretty much destroys the population of T helper cells. Okay? The CD4 marker is the thing that HIV uses to recognize its appropriate host cells. Okay, and when the virus destroys your T helper cells, then you're not able to do that. You're not able to do this whole thing of adaptive immunity, right? And so one of the hallmark signs of AIDS is that things that would normally be taken care of by a relatively mundane adaptive immune response, things like Kaposi sarcoma or toxoplasmosis, these normally very innocuous kinds of minor challenges to the immune system become insurmountable because we don't have the T helper cells to assist ourselves to launch the appropriate immune response. All right. I know, long video, but it is the last one, and I would normally have split this up into two. So consider yourself free from that last lecture video from Unit 14. Okay, bye.